Thank you, Edward, and welcome everyone for this Reaccess per Jisc training on how to advance your chemistry research through a simple and streamlined workflow. My name is Giulia Moncelsi. I'm a customer consultant in life sciences at Elsevier, and as such, I provide dedicated training and support on databases such as Reaccess, Reaccess Medicinal Chemistry, Embase Pharmapendium, and others. Please feel free to reach out to me via LinkedIn or Twitter, or you can also send me an email to this email address. So any means of contact that you prefer. So without further ado, I see that we're slightly over time, so let's get started. So what is Reaccess? Reaccess is the largest database for experimentally validated substance properties and reaction data, and it is presented with chemistry as the organizing principle. So overall, it is a powerful all-round research solution for facts and literature around chemistry and the broader chemical sciences. Our chemistry fundamentals span and comprehend over 118 million substance records with over 500 million excerpted facts on their properties. And the properties can be physical, chemical, spectral, ecological, all the way to bioactivity data. As for chemistry reaction content, we are happy to host around 49 million reaction records, and we do include excerpted data on reaction conditions, solvents, catalysts, yield, and all the technical aspects around an experimental procedure that you might be wanting to know more about. These chemistry fundamentals are contained in around 59 million document records from 16,000 journals, patents, and conference proceedings, which describe application in chemistry, so core chemistry, but they also span their uses across disciplines. So these titles span from content in material sciences, biomedicine, geosciences, engineering, and all the way to pharmacology and biochemistry, for example. So what makes Reaccess uniquely valuable for chemistry research? Well, from these 16,000 titles that I mentioned just a moment ago, we have two ways of performing excerption of the content. This can be done in two ways. It is both a manual and automatic process, and it allows us to extract within Reaccess the bibliographic information, the relevant chemistry concepts, the relevant chemical substances, their properties, and reaction data, as I mentioned. So all this literature is contained in our Elsevier document repository. And for the most part of these titles, we perform an automatic processing, which allows us to extract, as I said, the bibliographic information, concepts, and substances. On top of that, for 421 of these titles, so for 421 journals, we also perform a manual processing of the data to be able to extract with the highest accuracy and precision the chemical substances, the reactions, and other relevant facts around the information contained in those journals. So overall, this allows us to provide the relevant answers to key research questions that a chemist might have. For example, what properties have been reported on this compound? Or what substances are known to exhibit this property? Should I buy or make a specific chemical compound? And have similar substances been used in this way? Or can this substance be used, for example, for drug development application? So our excerpted data aims to provide high-quality, actionable experimental information. This is just a slide that recaps what we just talked about. Reaccess excerpts and quickly retrieves the most relevant chemical information. And the data, how does the excerption process work? So the data is selected from journals and patent literature, and it is systematically entered as consistent data sets in predefined masks of an appropriate excerption interface. So as you can see here, we have our bibliographic record, And through the extraction process, we are able to extract chemical structures, chemical synthesis, physicochemical properties, and pharmacology. I want to mention that for substance properties, these are not displayed as spectra or as images, but we do extract all the information in a tabular manner. So everything that you need to know is listed in tables, and this happens in all the sections across Reaccess platform. And this allows us to be as precise as possible and to be as consistent as possible as well in the information that we provide. And of course, from these records, we also extract all the relevant index terms and keywords. If you would like to learn more about Reaccess or how to use Reaccess to learn chemistry, we have just recently launched a very nice YouTube playlist called Reaccess Education, or RxED. And the playlist contains videos from the Reaccess Support Center that teach how to use Reaccess. And for this, you will find videos that start with Learn Reaccess, but also videos that are core content focused on how to use Reaccess to better learn chemistry. And these start with the words learn chemistry. So this is the link, the YouTube link. We will provide the slides of the presentation in PDF format, so you will have all the active links that are needed to get access to the playlist and other resources. Among other training resources and learning resources, we also have the Reaction Flash mobile app. So this is a mobile app that's available for any kind of phone, so for Android or iOS operating systems. And this was developed in collaboration with Professor Carrera, working at the ETH Zurich. And Reaction Flash gives access to over 1,000 name reactions, comprising their mechanisms and relevant examples published in peer-reviewed literature. Now I would like to start with displaying the Reaccess key developments that we had this year in 2021. We had substantial enhancements in our commercial substances, the patent content expansion within Reaccess. We also have a new synthesis planner interface, which is coming soon live in Reaccess. And something that just came live is our integration of Reaccess substances into Scopus. So the first big improvement that happened in 2021 in Reaccess is a more direct and easier access to results from patents. So now the patent content can be searched and viewed in Reaccess through the normal searching routes. For example, if you if you do a simple keyword search by topic, and then you go in the results page and you skim through the documents, and then you refine by document type patents, then you will have something that looks like this. The main enhancements of the patent expansion project are discoverable and viewable directly on the document results page. So for example, the title, these improvements are visible and coming from bibliographic enrichment. So for example, all that is about the title, the bibliographic reference, the abstract claims, and the front page information for a patent. And then we also have hit substances that come from the substance enrichment. And if the patent is about biochemistry or includes bioactivity data, we also have target enrichment information available there, for example, within the index terms. I have also summarized the main improvements that this patent content expansion project has brought to Reaccess. So we are building the largest collections of patents relevant to the pharmaceutical and chemical research. There's several items that I would like to go through. For example, we have access to claims from 105 patent offices and 170 IPC classes that allow Reaccess users to stay updated with the latest chemistry patent data information, especially in a time where joint industry academic partnerships are at the heart of the academia to industry transfer. You will also be able to discover non-English patents via keyword search with the title, abstracts, and claims translated to English. You will also have access to target and substance data from the 12 major patent offices, and this might help identify emerging competitors for a certain research project. And finally, you will have a fast turnaround with just 5 days from the publication of the patent to its inclusion in Reaccess. So you can stay on top of new developments. And at the same time, we aim to provide highest quality indexing in the shortest time frame possible. The second main improvement that we have in Reaccess is our synthesis planner that's coming soon live in Reaccess, possibly by the end of August, but not too long from now. So we improved and combined the user interfaces for synthesis planner to offer a streamlined experience with more relevant options for published molecules. This is how the new synthesis planner starting page looks like, and it will include the inbuilt functionality to draw compounds or reaction steps, which was not available before. So you had to build your search either in Quick Search or Query Builder, and then from the results page and from the resulting substances or reaction, open your query within the synthesis planner. Now this is being avoided because we have the possibility to draw your molecule or reaction directly into the synthesis planner. We also offer a route overview, including past searches, which was also not available before. So you will have what we call synthesis project window, where you can see the synthesis routes that you've searched for in the past weeks or days or months. And all the published reactions that you find within Synthesis Planner will be available in one seamless display, including literature references, experimental conditions, and published reactions. And finally, you can export the selected results in multiple formats. Supported formats are, for example, PDF, XML, doc, and SD files. So these are just a few more snapshots on the specific improvements to the synthesis planner that I've just commented on. We also have the predictive retrosynthesis Reaccess module, which is optional, and if purchased, it can be integrated within the synthesis planner interface directly in Reaccess. So you have one main interface to search for either published or predictive synthetic routes. So this is, as I mentioned, the starting page of the new synthesis planner. You have the inbuilt drawing functionality with our structure editors, Marvin or ChemDraw, that allows you to draw molecules or synthetic steps. Then here you will have your chemical routes and synthetic routes overview. Here you are seeing the tree view, and that allows you to easily navigate Reaccess examples and conditions. And finally, as I mentioned, you have different exporting options that allow you for more flexibility when transferring the knowledge accumulated on Reaccess to your day-to-day research in the lab. Another major improvement that just came live now in Reaccess is that if you have access to Scopus, now you have direct access to some of the substance information that's powered by Reaccess database. So you will have ultimately a very enriched search experience. The new features include a revamp of the Scopus document page with a redesigned Reaccess section containing a carousel navigation that allows users to quickly scroll through relevant substances directly in Scopus. And if you click on any of the substances, you will open a flyout containing chemistry details from Reaccess, including the types of data available for specific substances, so spectral properties of physical data or bioactivity. And finally, and most importantly, you always are presented with a direct link to the corresponding data in Reaccess for each data type, including, for example, links to commercial suppliers, drug likeness, and preparation information. We also made substantial improvements to commercial supplier information, and we're extremely happy to say that Reaccess now includes around 400 commercial suppliers and growing, with a dedicated searching environment. So when you perform, for example, a quick search and you are in the preview page of results, you can see the different environments between substance searching and then commercial substance searching. In the next releases, we will also have a shopping list kind of feature that looks like a one-stop shop that allows users to seamlessly collect commercially available products via Reaccess, and these products will be added to the list from different searches, and you can review the list as you go on purchasing. So you don't need to go to the different suppliers' web pages to even compare the chemical information or the price or the availability of these compounds. So this was about the initial introduction to Reaccess and the main developments that we had this year. And now I'm really happy to present a researcher's workflow on how to accelerate it from the idea generation to the execution of the experiments in the lab, and hopefully finally to the publication process. And we don't have Dr. Ricardo Di Sanza available live with us today, but we have recorded a 20-minute session just one day ago. And so we will be broadcasting that one. So Dr. Ricardo Di Sanza is a senior scientist at Signature Discovery, a CRO based in Nottingham, UK. And you can reach out to him via LinkedIn or Twitter if you're interested in knowing more about his research.
Okay, thank you. Well, hello everyone. Thank you, Giulia, for the kind invitation. I'm very pleased to present this project that I've been involved during my PhD at the University of Nottingham. And to be honest, I've been using Reaccess during my graduate studies a lot, and I'm very pleased to present and this training about Reaccess because it's been very useful during my studies. And as I said, it helped me very a lot in overcoming some problems during research that I have undertaken as a PhD student. So I'll start by introducing myself. I'm Ricardo, I'm Italian. And after studying for my undergrad degree in Italy, I moved for my postgraduate studies at the University of Nottingham, where I studied in the group of Professor Hong-Wai Lam in the field of enantioselective transition metal catalysis. After being awarded the PhD in chemistry, I moved to Tarragona in Spain at the ICIQ, where I worked with Professor Paolo Melchiorre in enantioselective photocatalysis. And finally, I moved to my current position, where I work as a senior scientist in drug discovery at Signature Discovery, a CRO based in Nottingham. So during this presentation, I will show you how I used Reaccess for one of the projects that I was involved into during my PhD. So during this, at the outset of this project, I was discussing with Hon, my supervisor, how to perform some enantioselective transformation to aim to synthesize a core structure found in biological active compounds. Now we started to do some brainstorming, and we came up with some ideas. One of the possible ideas was this, which was pretty feasible in our mind, since involved the formation of a lactam, and we all know that lactams are widely found in biological active structures. through the formation of these allyl metal species that could undergo a nucleophilic allylation. So before starting to go into the lab and perform some reactions, the first thing that I did was go into Reaccess and start to build up my my knowledge on the topic of nucleophilic allylation. So what I did, went to Reaccess, quick search, type in an anti-selective nucleophilic allylation and other searches that could come up to my mind relative to this topic. And then I will find different hits that will be of different documents that will give me a background on this topic here. Of course, I couldn't read all of the papers found, but I refined the search by review, and I chose few reviews that based on the number of citations that they had and how recent they were. So I choose few reviews, like 2-3 reviews for starter, read them, digest them, and then start to look to the papers that the review cited and the papers that cited the review, always using Reaccess. So once I found this, I built up some knowledge on the topic. I checked whether the idea that we had on the methodology was previously reported. And there was just, with this search on Reaccess, I found out that there was just this report by the Tsukamoto group in 2008. They reported this nucleophilic allylation quite similar to the one that we had in mind. And they had this cyclization with high levels of enantioselectivity and great yields. However, there was one caveat with this chemistry, is that this worked very well with aldehydes, but less with ketones. As a matter of fact, when they reported the only ketone present in the scope, they had a massive drop in enantioselectivity and yield, and they had to use harsher reaction conditions. So with this background search done by Reaccess, I could find a potential way to expand this kind of chemistry. And in fact, my fundamental questions that I wanted to answer were, can we expand this reactivity to ketones, always having outstanding levels of enantioselectivity and yields? And plus, can we use potentially a metal as a catalyst, but that is which is more earth abundant than palladium? Furthermore, what I wanted in the beginning, well, my supervisor was very keen on not only expanding or like finding very cutting edge research, but even to apply this research to synthesizing some useful molecules, so biological active compounds or core structure found in biological active compounds. So what I did was to go back to Reaccess, draw the molecule that, well, the core structure that I had in mind, this gamma lactam that has a tertiary alcohol alpha to the lactam. Now, with some research tools that are present in Reaccess, I managed to, well, this S6 just means that in these positions that I customized, we can have any sort of substitution pattern. And so it will give me a wide array of compounds bearing this kind of molecules, but these molecules will be changed only in the positions that I wanted. So once I performed this kind of research, as before, I had some hits, and thanks to those, I could find that this kind of gamma lactam is quite useful because there are actually patents that describe this gamma lactam with an alcohol here that has potential to be an antidepressant, an anti-angiogenesis, or an herbicide. So now that was it. Like I had the idea of how the reaction should perform. There was potential to expand some chemistry and do some totally different, like to expand even further some chemistry. And we had a target in mind, which was this gamma lactam, which is very useful. So with this heat, the only thing that I had to find out now is how to synthesize this compound, this starting material. As well as, again, I went back to Reaccess and I started to find out ways to make, for example, this alene. And the way I will do is go to quick search, reaction search, and then draw the molecule that I wanted, with drawing the arrow on the left-hand side, so that I will have all the ways possible to synthesize this molecule without limiting myself to any specific starting material. So I will get some heats as before, and of course there are several reactions that could form alenes like this, and with some experience in hand, I will choose which are the reactions that will be useful to pursue. So once I found them, I will go back. So in this case, I will have an alkyne. The alkyne, doing some retrosynthetic analysis, I will go back and using Reaccess to find the most viable synthesis, which led this three-step synthesis of this alene. That is my starting material I want to start it from for my reaction. So that's it. We have the heat, the target, we have the reaction that could be improved, and we have the starting material done thanks to Reaccess, thanks to the help of Reaccess. So we started to look into reaction conditions, which I will avoid for the interest of time. And we found out that this kind of chemistry could be performed with nickel as the catalyst, way more earth abundant metal compared to palladium, in combination with these FOX ligands, and in very low catalyst yields can perform the nucleophilic allylations of these ketones tethered to alenes. In quantitative yields, only one observable diastereoisomer and outstanding enantioselectivity, selectivity, plus without the need of any purification. So we started going on with the substrate scope, with the boronic acid scope. These are just two highlights of the boronic acid scope that we performed. Very important pieces, and above all, this one here with the fluorine atom in the ortho position of the boronic acid, which allowed us to have a very nice crystal structure that confirmed the enantiomer that we were getting. Now, considering the fact that this is an ortho-substituted boronic acid, there is something else to say. Well, as I said, this was done. This kind of research was done during my PhD, and the University of Nottingham organized some events where students could present their research in front of, excuse me, in front of academics and industrial partners. So I presented my research and I received very outstanding questions. But two of those questions were amazing. And they were asked by Dr. Liam Ball, an academic at the University of Nottingham. He basically asked me, once he saw the boronic acid substituted in ortho with a fluorine, he asked me whether this chemistry will work with boronic acids with an ortho substituent which is bigger than a fluorine, because fluorine is very small. It's kind of cheating in a way. It's not, but it's kind of. And the second one is, does this chemistry work with nitrogen containing boronic acid? Now, imagine I was presenting in front of everyone, in front of all the academics, all my fellow students, in front of industry partners, and he actually found Liam found a blank where I didn't work on. So the first thing that came up to my mind was, he got me. But of course, like after this alpha second of panic, I actually was trained by my mentor, Hon, to say out loud, well, this is a very interesting question. I didn't think of that. So without the other jokes, like the thing is, these kind of questions, as I said, were amazing. They tickled my curiosity and motivated me to go back to the lab and try to test this hypothesis. So what I did, I started with the orthosubstituted boronic acid, and I was very pleased to find out that this chemistry is actually tolerant to orthosubstituted boronic acids. However, regarding the second question, which was if this chemistry is tolerant of nitrogen-containing boronic acids, it wasn't very so straightforward happy days. In fact, when I tried all these boronic acids with nitrogen, they all failed. Nothing happened. I recovered only starting material. Now, I was kind of bothered about this, so I started to think about ways why this didn't happen, like this chemistry didn't work with these specific boronic acids. And I could think of two possible answers. Either that these boronic acids are not very stable in the reaction conditions, or that this kind of nitrogen functionalities hamper the reactivity of the nickel catalyst and takes it out of the catalytic cycle and then shuts down the reactivity of the catalyst, so there's no reaction going on. So to understand which hypothesis was in place, I went back to Reaccess. I did a topic search exactly as I showed you before, checking different things, and above all, like how to perform like a screening on how to see how robust a reaction is. And I found out this paper by Glorious, where his group describes this robustness screening, which is basically run the model reaction with a normal boronic acid and try to add some additives and check if you can get exactly the same result in the first place and how much additive is left, to check if this additive, the nitrogen in my case, was hampering the reactivity of the nickel. And I was pleased to find out that the reactivity wasn't hampered at all, and I could recover all the additive that I added. So possibly what was happening is not that the nickel wasn't active anymore, but probably that the boronic acid with the nitrogen wasn't very stable and wasn't surviving the reaction conditions. So I was starting to think how to come around this way to get some boronic acid with a nitrogen containing a nitrogen in it to work. And well, this is the slide that I showed you before, where I was pleased to find out that this chemistry is tolerant of ortho-substituted boronic acids. And I thought probably since this works with ortho-substituted boronic acid, the nitrogen can be placed in the same position and could potentially, maybe, stabilize the boronic acid for just a little bit to allow the reaction to take place. And as a matter of fact, well, this might be the answer. In fact, putting a nitrogen next to the boronic acid allowed us to have this compound here in moderate yield, only one observable diester isomer, and modest enantioselectivities. But still, for me, was a good proof of concept. Then we are not limited just to gamma lactam. We could form other pyrrolidines as well, starting from this starting material that required different synthetic pathways compared to the model one. So I went back to Reaccess, placed the research query, and then find out how to synthesize this other starting material. Same thing for all carbon containing starting material, both 5 or 6 member rings that required a totally different approach to synthesize the starting material, and with the help of Reaccess, I managed to do that. Further to this, even piperidines, and starting with the starting material, Reaccess helped a lot as well. Now, all these experiments done, and the experience with these experiments, plus the fact that in the beginning I had this literature search that built up my knowledge of the topic, we could come up with a mechanism, a possible mechanism for this reaction. So starting with the active form of the catalyst, which then can perform a transmetallation with the boronic acid to form these organometallic species, which then can react with the allene through a migratory insertion with the aid of this directing group. Then a sigma pi sigma isomerization, which lead to the formation of these allyl nickel species that can react through a Zimmermann-Traxler transition state with the ketone. That explains the high levels of diester selectivity always encountered in all the reactions. Plus, concerning the enantiomer selectivity, the possibility is that the phenol group is on the other side of the terbutyl present on the ligand of the nickel catalyst, which explains the high levels of enantiomer selectivity. We will have then an nickel alkoxide group that then will be liberated by a proton source to close the catalytic cycle. Now, this is it. I thank you all for the kind attention. I hope I made myself clear how I would use Reaccess on the day-to-day basis. Of course, these are really basic functionalities of Reaccess that I use, as I said, in the day-to-day basis. But there are other more in-depth features of this search engine that I would use, but not as frequently, but still very useful. And as I said, I used it a lot during my PhD and helped me very much. So thank you all for your kind attention, and thank you, Giulia, for inviting me for this training.
Great. And although he's not live with us today, I would like to thank immensely Ricardo for his contribution to this Reaccess training. And you can see, I hope you could see from the recording and from his talk how he used Reaccess in each step of the way on the path to publication of an idea. So here I have summarized the path from the idea generation and the birth of a research project in chemistry, all the way to the synthesis and preparation of new or already existing and published substances that can be then used for that can be used in developing the research projects, in finding, for example, new synthetic methodology, as Ricardo did for his article published in Chemical Science last year, or to just use the Reaccess data to formulate new applications, and just in any way to do the reporting and publication and dissemination of the results from the idea of the research project. And so this is like a very useful workflow to tackle the proposal, design, the execution, the assessment, and the dissemination of the results that you have obtained, also thanks to the help of Reaccess, for example. So I hope this is clear enough, and I hope that by now you could grasp some of the additional benefits that using a search engine like Reaccess can be the chemist and to their daily work in the lab. But also, I want to mention that Reaccess can be used in all fields of chemistry. So we've seen with Ricardo's example in catalysis and synthetic methodology, but really Reaccess is an invaluable asset. Also, if you're a material scientist, if you're working with biologic applications or nanotechnology, all the way to theoretical chemistry. So just to have access of such a breadth of data and substances and reactions and their properties is of great value and can really help you all the steps from the idea generation to publication and dissemination of your research in the chemistry community. So I would encourage you to think of all of this when you're using Reaccess and not considering just a chemistry database, but a tool that can help you in all the different steps of your day-to-day work. So now I will move on to the last part of the presentation. I have calculated that this will take around 10 minutes, so we will finish around 1:30 as planned, but then I will ask all the attendees to stay for an additional 5 to 10 minutes so we can address any questions in the Q&A. And there will be also some questions that will be prompted to you. So if you could answer that, that would be fantastic. This last section of the presentation is focused on the Reaccess predictive retrosynthesis module. So now, as for the initial introduction, retrosynthesis, what is the need for it? And how can I make new compounds and new molecules using Reaccess today? So as most of us know, retrosynthesis is, I would say, an art in chemistry, in the chemical scientist, to be able to think of the fastest and less expensive and easiest route to fabricate a certain molecule that hasn't been published or made yet. And this is done by having the final molecule and then thinking retrospectively and retrosynthetically to all the steps that are needed to make that molecule. And with this process of thinking, then you come up with the initial starting materials or reagents that can be either commercially that can be purchased commercially or can be made in a really easy way in the laboratory. So how can you do the retrosynthetic process or make new compounds using Reaccess today? Well, currently, using Reaccess, this translates into a 4-step process. As comparison, we will compare this process to being in 2021, and instead of using navigators, GPS, and digital systems, we would still be using a road atlas, for example. So a book that explains to you, that showcases the maps to how to reach from point A to point B. And this process is divided in, as I mentioned, in 4 steps. First is thinking about the chemistry needed and the steps needed to make a new molecule. Then the second and most cumbersome process is the extensive reaction and literature search. Then once the the literature has been skimmed and you found the peer-reviewed and published literature data, you can start your reagents and starting material search, and finally you can translate all of this process to the lab, so the work at the bench step of the process. And within Reaccess, this happens when you try to synthesize a new molecule, that such as the one that is depicted here, and you launch the search, and you're coming across this kind of scenario where you have 0 results in the substances and reaction data. So you could overcome this problem by allowing for substructures and allowing for more modification to the molecular backbone, but you will not be able to search to find the synthetic route for that specific new molecule because it hasn't been published in the literature. So the answer to this cumbersome process is through Reaccess is to use the new Reaccess predictive retrosynthesis module. And the question is, how can I make a new compound using this module in the future? We will briefly see how the predictive retrosynthesis module has been constructed and what is it, the underlying AI and machine learning technology that it has, and how it is built in a way that it learns from the existing literature, chemistry literature published, and how it can provide the answers and how it can predict the synthesis for new molecules. But overall, in this slide, I want to point out that through this predictive retrosynthesis module, you will be able to reduce the process that we've seen before from 4 steps to 2 steps. So you will be able to just first of all, think about the chemistry again. So this is very important in the sense that the figure of the chemist will not be substituted by the algorithm or the artificial intelligence from the predictive retrosynthesis module, because the chemist will still have to think on the feasibility, the viability of the route proposed and predictive, and also the chemist will always have the option to choose their own routes if they have been already tested and vetted in their own laboratory. And finally, the second step will be, of course, translating this knowledge acquired through own thinking and the use of the predictive retrosynthesis to the lab as before. I just want to mention how Reaccess retrosynthesis works in a nutshell. So it combines Reaccess reaction knowledge base. So for the time being, around 15 million single-step reactions of small organic molecules were taken from Reaccess database, and around 400,000 reaction rules were extracted from these chemical transformations. And then all this knowledge was fed through the underlying technology of the predictive retrosynthesis, which are 3NMCTS, or 3 neural networks coupled to a Monte Carlo tree search. And all of this results in the final user interface that you have in the Reaccess predictive retrosynthesis. This is, for example, is a snippet of a tree view of a predictive synthetic route. And everything about the algorithm is, or the most important things to learn about the algorithm were published in this seminal Nature paper 3 years ago by Professor Segler and Professor Woller, which are our collaborators to develop the predictive retrosynthesis module. So again, we try to all the time meet your needs through Reaccess and beyond. So whenever you come across such a scenario in Reaccess for which no substances or targets or reactions are found, meaning that your molecule hasn't been published yet, then you can predict its synthesis to the retrosynthetic predictive retrosynthesis module. And again, here a snippet of the tree view of a particular predicted synthetic route in the module. And you can switch from tree view to table view as you have available in when you find published reactions in Reaccess. So it really works in a very similar manner. And I want to anticipate that it's really straightforward and easy to use, because let's say all the computing and thinking work is being done on the background by the AI, and then what is presented in the interface is just very straightforward. One practical example where we can use the predictive retrosynthesis module, for example, Pfizer has just recently started clinical trials for this molecule that's used as an oral antiviral against COVID-19. And there's a reference of the beginning of the clinical trials with this molecule, but of course we didn't have any detail on how the molecule was produced in the lab by Pfizer. So what we did was we processed it through the Reaccess predictive retrosynthesis tool, and here on the right hand side of the slide you can see a snapshot of how the synthesis has been predicted. And we have also like summarized the main synthetic steps. And these are all very feasible transformations. So this is just one example on the reliability of this tool. Of course, it always needs to be double checked by the expert eye and mind of a chemist, but overall, we can already ensure a very, very high reliability on the predicted routes. As such, as kind of a confirmation of that, the Reaccess Predictive Retrosynthesis won the Global Life Sciences Award 2020 for the best chemistry predictive analytics specialist last year. And so this is just one of the recognition that we've had for this module. So just so you know, and finally, in this final slide, I condensed the take-home messages and what's important to remember about the predictive retrosynthesis. So first of all, about the AI and machine learning training datasets, the predictive algorithm was trained on the largest chemistry reaction dataset from Reaccess with this amount of auto-structured rules and virtual negative chemical reactions. So we also took into account when reactions are not working. And this is extremely important because it helps the algorithm learn more. And this is also something that a chemist would see on a day-to-day basis in the lab. We have had collaborations in the pharma and chemical industries from very renowned partners. As for the technology, as I mentioned, you need to remember that it's powered by 3 neural networks coupled with a Monte Carlo tree search. And it's a technology that enables to easily update the model so that predictions are fast-paced, such as chemistry innovation. The third point is extremely important. We confirm that this module is scientifically robust, and it's been tested using 3 different approaches. For example, rating the false positive rate, or the time split validation, indicating that the model performed with high accuracy and speed when analyzing many molecules at the time. So it's also important to have fast response from a module. It currently is wired to produce an answer within 2 and 30 minutes. So I think this is an excellent time frame for such a module. And finally, we also perform chemical Turing tests by testing it with many organic chemists worldwide that indicated high quality and robustness of predicted routes versus the ones that they thought of manually. And yeah, you can if you access the module, you will be also be able to customize it by adding own building block libraries from a university or a repository of such molecules. And you can also augment it by adding own chemistry reaction data. And if you would like to know more, we ran a general webinar on this new module, and you can still register to watch the recording on demand. The slides of this presentation will be available in PDF to you, and the active links are also available in there. So you will be able to access this webinar and register whenever you please. Thank you. So I see that we're quite on time, and I think that we can go live with the Q&A right now. And I would just like to thank you all for attending, and the floor is open for any question. And I see that some poll questions that we had prepared are being posted. So if you could also answer those within this Q&A timeframe, that would be fantastic.
Thanks so much, Giulia. That was excellent. Yeah, as Giulia said, you can now post your questions in the Q&A, and you'll also see now that you've got mic access. So if you'd like to ask a question verbally, please go ahead. The fact that there's not too many people here means that you don't need to raise your hand. I think we'll be able to manage with the microphones just being enabled. So if you'd like to ask a question using your mic, go ahead. Thank you.
I've seen that there is one in the chat, and at the same time I turned off my video. I turned on to just say hi to everybody. Hope you can see me now. I have a question from Jisc. Thank you for the nice presentation. For the retrosynthesis module, does it suggest more than one route? Yes, it does. Thank you for the feedback, Jisc. Yes. So whenever you launch a search on the predictive module, and you define some parameters, for example, the diversity or the number of maximum synthesis plan or predicted routes that it can create. So you have these parameters that you can toggle to be able to define beforehand what you would like the module to predict. And then say, imagine an upper limit of 10 synthetic routes that are predicted by the module. And then you launch your search. It will predict them within the time frame that we mentioned, between 2 and 30 minutes. And then it will find the highest number of routes. It will predict the highest number of routes. So it could be any number between 0 and 10, for example. And for each route, you will be able to go through it in a table or tree view. And then for each route, you can see all the different steps. So, and there's some very easy breadcrumbs that you can follow to go back to the main, to the homepage of the module, where you have all the synthesis projects that you've launched in the same day, but also in the weeks or months before. So everything is stored in the platform, and you don't even need to save your search because it's automatically saved. And I want to mention that it is currently being integrated in Reaccess. So sooner rather than later, if you have both Reaccess and the Reaccess predictive retrosynthesis, you will be able to access the predictive retrosynthesis feature through the synthesis planner tab, which will be renamed as retrosynthesis. Which means that you have everything in one platform. So this is already a big advantage. And also within the retrosynthesis tab, you will be able to decide, if you subscribe to both, of course, decide what kind of result you want. If you want peer-reviewed and published literature retrosynthesis, or if you want just predicted routes. And of course, you can always combine this information to a later stage and use it for your research work. Right. Thank you, Luis. I see a follow-up question. How straightforward would it be to compare price and affordability of starting material? So for that, of course, you would need to rely on Reaccess. So if you already know which starting materials have been used or have been predicted by the retrosynthesis module, you can launch a structure search or a quick search through structure search on Reaccess. And then you will find if of course, if they're commercially available, as I mentioned, you will find two types of answers. You will find the substance data, but you also have say a preview environment of the commercial substance information. So if you expand on that, if you go to Reaccess and you expand the section about commercial substances, you will have a table view that lists different commercial suppliers in alphabetical order. So, for example, I don't know, Merck, and then you would have Fluorochem or Enamine or Merck, and everything is listed in alphabetical order. So the suppliers, and then you have different columns and rows. And in the columns, you can skim through the price, the purity, and the amount that it's sold on this particular supplier. We also have a fully integrated, for example, the e-molecules repository, and we also have lab networks and I think also Sigma Aldrich data. So you have everything in a tabular view. You can export that, and then you can compare. And of course, we also provide active link to the commercial supplier webpage. So if you want to purchase now, at the moment, you still need to go through the commercial supplier webpage. But in the future, we want to make it available to purchase directly from Reaccess. Okay, let me know if there are other questions. I see that the poll is being populated. We can give one more minute. We have one other question, the final question. Now I'm scrolling the chat to see if there are additional questions. Doesn't look like it.
Thank you, everyone. We do just have two final questions, general questions about this webinar. Let me just bring those up for you. So we'd really appreciate you filling in these last two polls before you leave, before we culminate. You should see them now on the screen. Thank you. Okay, I know we've run over, but obviously that was due to starting late. So Giulia, thank you for making that adjustment, and thank you for everybody for waiting so patiently and staying till the end. It's much appreciated.
Yeah,
might see.
I would like to thank you, Edward, for guiding us through the platform and for hosting the webinar. Thank you.
No problem. Thank you. It was a really good session, and I hope everybody that's attended has found it really beneficial and interesting. I mentioned at the start, which some of you may have missed, that it is being recorded, which I can now stop. Well, I'll stop in a second, and that will be made available on the webpage within a few days. So if there's anything you'd like to recap on, feel free, and we'll send a follow-up email with all the details on there, including the feedback form link. But I've also just put that in the chat pod, so we'd really appreciate your feedback. So if you have time to fill that in, it doesn't have to be now, but at a convenient time, that'd be again, much appreciated. So just leaves me to thank Giulia Moncelsi and Ricardo de Sanza for their presentations and for all of your contributions and of course for your attendance. So other than that, once you're ready to leave the session, all you need to do is click the room name at the top there where it says Jisc webinar, and you just go down to exit room. So thank you. Thanks, Giulia.
I echo Edward in thank you, Edward. Thank you very much, and thank you everyone for attending this training, and have a great rest of the day. Thank you.